Hi, my name is Dr. Scott, and I'm going to talk to you a little bit about the research interests that I have and things that I've done in the past. Generally, my philosophy for students is trying to give students the opportunity to research what they're interested in, so I'm always open to ideas that students might have. If I can't contribute, I'll try to find some way that my skills and your interests can combine. I generally have my students who are interested in researching take my 496 class. They take that and listen to the research projects and kind of learn about what's going on and contribute where they can. And if they find that they are being able to be contributive and effective, then they can continue with researching with the group. And that's something that a pattern that we want to use to kind of make sure people are really interested in doing the research and feel like they want to contribute to what we're doing. This is my homepage here on the BYU Hawaii website, and these are some of my research interests, or I guess some of the things that we've been working on over the years. Biofuel cells in general is probably the most abundant amount of research in terms of publications that come from our efforts. So in general, a hydrogen fuel cell utilizes hydrogen and oxygen to produce energy. This is somewhat of a problem because hydrogen is not readily available as a natural material, natural product, or natural resource. So the hydrogen fuel cells never really developed into something that is used more than just in very niche applications. A biofuel cell is a scenario where we're trying to replicate the hydrogen fuel cell using components that are more sustainable, using a biofuel, for example, glycerol, glucose, or ethanol. These are things that we've tried, and it can be enzymatic. Glucose is where we've made lots of our strides, and oftentimes the process has to do with mediated oxidation of glucose. So a small molecule mediator accepts the electrons from glucose and then can pass those electrons onto a surface, because glucose isn't very good at oxidizing cleanly at any given surface. Okay, so our first publication on this paper was in 2009, where we looked at the resulting fuel cell that we created. This is a little mini fuel cell where we have glucose oxidizing at the anode, and the cathode reduces oxygen to the hydroxide ion. Okay, and we can characterize the fuel cells and see how well they perform. We can do mechanistic studies with different compounds and try to identify what the mechanism for the processes are. We try different mediators, different materials that will accept electrons from glucose and pass those electrons onto a surface. And we've done all sorts of characterizations of the cells to try to, again, identify what the mechanism for the reaction is. All right. Another more recent area of research that we've been interested in is anti-cancer properties of beta amyloid. So we have a cell line, lung cancer cell line, that we can expose to different compounds and see how effective those compounds are at preventing cancer growth, right? This is a recent paper that was presented by a couple of our students, where they were looking at the effect of amyloid beta protein. Beta amyloid protein is a protein that's found in Alzheimer's patients. It's a protein that we're learning more about, not just us, but the world in general. And beta amyloid seems to have the ability to kill microbial and viral antigens. And the question was, how would it interact with fast-growing cells? And so we're doing a little bit of study there, and we found some interesting stuff, and we're interested in looking at mutations of amyloid beta and trying to do the same types of experiments. I'm also interested in tungsten carbide corrosion and oxidation. done a handful of experiments with with a company from Orem, Utah, and we found some really interesting results. And it's a very important application for the oil drilling community. So that's another area that I'm interested in working on. Bacterial identification. This just generally means kind of understanding complex bacterial colonies, but samples, biological bacterial samples, using the liquid chromatography mass spectrometer, the LCMS. So here's an example of a poster where we were looking at how the LCMS could be used to identify complex mixtures. So using known bacteria, combining them together and mixing them together, and kind of watching how these bacteria would have wars with each other and determine who would end up being more prominent, and seeing if the mass spectrometer could kind of identify that for us so that we could perform experiments. For example, we have a student who is washing their hands with different soaps and then trying to see what kind of bacteria remain, and if that bacteria begins to predominate on certain people's hands based on kinds of soaps that they use, because there seems to be more and more evidence that suggests that something about our environment is causing our gut bacteria to be altered, and that this is resulting in lots of different disorders and diseases. So this is something that I'm interested in, trying to look at general bacterial samples and changes of bacterial colonies and surfaces in general, just on more of a macroscopic perspective. We have been making soap for the cafeteria here on BYU Hawaii for pushing 7-8 years now, and it's been a great experience. Students who do this use the waste vegetable oil from the cafeteria, turn it into soap, hand soap, and then deliver it back to the cafeteria. And it teaches us a lot about sustainability. It also allows us to use chemistry to pay our way through college. So students who do this generally will have a job the whole time they're here at the university. And if people are, we've always been kind of promoting this a little bit because the university doesn't use this soap. It's only the cafeteria on the university, right? So the whole university could be using it if there was somebody to kind of spearhead that operation and take it to the next step. So we're always interested in trying to see if somebody could do that for us. Like I said, I've generally tried to find things that students are interested in, and so a lot of people are thinking about going into dental areas, the dental profession at some point. And one thing that I'm very interested in is how the influence of different bacterial colonies in your mouth, what the influence those different bacterial colonies in your mouth would have on your oral hygiene, right? So this is an example of an experiment that one student was working on for a while. We were taking dental samples, comparing the oral hygiene of the person to the bacteria that was prevalent in there. Or no, this wasn't the bacteria, sorry, this was the saliva proteins themselves, trying to find what the genetic component is that results in people having nicer teeth than others, right? Another example of work that we did, this is more of a natural products work, but I'm open to working on kind of this side, not in the building of natural products, but the classification. I think that Dr. Cornwall would probably be a better choice for this, but this is something that we've done in the past, just a little bit of characterization. We already talked about that one. The other one is the other area of work that I'm really interested in getting started in is what we call Moths, metal organic frameworks. Metal organic frameworks have this let me see here. Hold on just a second. There we go. You can see it here. Metal organic frameworks are a metal indicated by this red dot here, organic linker, and they come together to form crystals. Here's an example of our organic linker, can bind to 3 different metals of these carboxyl groups. And there's our metal. And the result is really cool, complex structures. And on the Big Island, we just helped. They did most of the work. We just did a little bit. But because of our little contribution, we're able to have access to an instrument that can solve crystal structures for us. And so we can create these moths. They do all sorts of amazing things. They can purify liquids, purify gases. They can act as drug delivery components, right? They're showing promise in all sorts of things. And they're relatively easy to make initially and very easy to make variations of. So I thought it was a project that would be very good for undergraduate students. So and that's about it. Again, there's other things that I would be interested in doing, but these are the things that we've been working on in the past, and I'm excited to move forward with these things. Again, I'll take anybody, but I really want to see students who are interested in sticking with research. If you're not interested, then after one semester of taking the 496 class, you probably just move on to something else. But okay, thank you very much.